Okay, so in chapter 9, we're just going to talk about how we control microorganisms in different settings. So when we're talking about microbial control, we're not just talking about killing germs or killing bacteria. What we're talking about are the methods. So what methods are appropriate for what purposes? And there's different techniques that can be applied. But regardless, these techniques are going to either completely remove the pathogen, they're going to inhibit the microbial growth, or just flat out kill or destroy the microorganism. So we're going to start off by talking about some terminology. These terminology are important because they all mean something different. So they're not interchangeable. So when we're talking about antiseptics, we're talking about or referring to the reduction of microorganisms and viruses, particularly pathogens, found on living tissue. So a good example is when the skin is prepared with either iodine or alcohol before receiving an injection. And when we're talking about an antiseptic, it really is just a diluted disinfectant that has been formulated to be safe for living tissue. We also have aseptic. So if we're using the term aseptic, this can describe procedures or environments. So if we're talking about an aseptic environment, a good example is a surgical field. If we're talking about an aseptic procedure, well, you do that in lab all the time. So when you flame loop sterilize before you transfer bacteria to a media, that's an example of aseptic technique, which you should know. We also have two important suffixes, so we have side and sidal. These indicate the destruction of a specific type of microbe. So some examples include bactericide. So this means that we're talking about destroying bacteria. Fungicides destroy fungi, so on and so forth. So we have virucides and germicides as well. So germicides can include chemicals like ethylene oxide, propylene oxide, and aldehydes, and these are usually used for high-level disinfection or sterilization processes. Okay, we also have degerming. So this is referring to the removal of microbes through mechanical means. So things like hand washing or alcohol swabbing at an injection site. So before, when we were talking about alcohol swabbing and iodine application, we're really talking about the chemical. The chemical actually only plays a secondary role. It's actually the physical action that's the most important. So when we're talking about degerming, we're talking about the physical action of alcohol swabbing. Then we have disinfection. So disinfection is the destruction of most microorganisms and viruses, but on non-living tissue. So we can do so with things like phenolics, alcohols, aldehydes, or even soaps, and apply these to equipments and different surfaces. It is focused on pathogens, but disinfectants do not kill everything. We also have pasteurization. We talked about this process, I believe, in exam 1 with Louis Pasteur. So it's going to be a process that uses heat to kill pathogens to help reduce food from spoiling and beverages from spoiling, so things like milk or juice. So the heat is usually only applied for a brief period, because if it's applied longer than that, then it would potentially alter the taste or the nutrient content. So because we only apply the heat very briefly, it does not sterilize. So some microbes are going to remain, and they can eventually spoil the product if it's not stored properly. Then we have sanitation. This is going to involve removing pathogens from objects to help meet public health standards. So a practical example is washing tableware in very hot water in restaurants. It is regulated, but the standards do vary between different jurisdictions. So this is why sanitizing something at home may not meet the same criteria as that for like a restaurant or a hospital. Some more suffixes. So we have stasis and static. These are indicating inhibition of microbial growth, but not complete destruction. So we have things like refrigeration, freezing, or even some chemicals. These can slow or halt the growth, but they don't actually kill the microbe. So one example is bacteriostatic agents that inhibit bacterial growth. So this is going to allow food or biological materials to be preserved until further processing. If the conditions for the bacteria improve, then the microbes can also resume their growth. And then finally, we have sterilization. This is referring to the complete destructions of all microorganisms and viruses that are on or in an object. So it's typically achieved through methods like steam under pressure, incineration, or ethylene oxide gas. So examples include sterilizing microbiological media, surgical instruments, or even the process of canning food. When we're talking about sterilization, this is the highest level of microbial control, and it's very important and it's crucial when we're talking about the medical field or even research settings where we don't want any viable organisms to remain. Okay, so microbial death, it doesn't occur all at once. When a microbial agent is applied, microbes are going to die at a constant rate over time. So the most susceptible cells are going to die first, and what they leave behind are the more resistant ones that take a little bit longer to eliminate. So this is a predictable decline, and it is represented by a straight line on a semi-logarithmic graph, which you can see here. So what this is showing you is that if 90% of a population is killed in 1 minute, then another 90% of the remainder population is killed in the next minute, and then so on and so forth. So this is why sterilization requires both a sufficient amount of time and the correct or appropriate intensity. So if we shorten the exposure or lower the treatment strength, that could reduce the microbial load, but it's not going to make sure that we have complete sterilization. So this principle is really important when we're considering cycles for autoclaves, how long a disinfectant needs to be applied before it achieves the expected death rate, or even pasteurization protocols in both lab and industry settings. Okay, so antimicrobial agents, they work through two main mechanisms. The first is altering cell walls and membranes. The other one is damaging proteins and nucleic acids. So if you remember the structure of a lot of microbes, they have a cell wall. So the cell wall provides both, helps with the structure and protection from osmotic pressure. So if the cell wall is damaged, the cell can lyse or burst. When we're talking about the cytoplasmic membrane, it acts like a selective barrier. So when it's compromised, the contents can kind of leak out and the cell will die. If we're talking about viruses, damaging the envelope is going to help prevent attachment and its replication, because then it can't enter into host cells. And it makes envelope viruses more susceptible to disinfectants than non-enveloped ones. And I think we talked about that in the virus chapter. Okay, the second mechanism, damaging proteins and nucleic acids. So proteins rely on their three-dimensional structure in order to function. So things like heat, radiation, and chemicals, they can denature these proteins because they are held together in their three-dimensional structure by hydrogen bonds. So these are weak bonds. DNA and RNA can also be destroyed or mutated. So this can stop protein synthesis and replication entirely. And we also have ribozymes. So these are RNA-based enzymes, and these are essential in translation. So if we have damage to nucleic acids, it can be fatal to the cell. Different agents work through different pathways. So alcohol and detergents primarily target membranes. We have oxidizing agents and radiation. These are going to damage nucleic acids. And then we have heat, and heat can damage both. So we really need to go in and talk about what these mechanisms are, and to make sure you understand what the right control